Can you hear us now?
Professor, you're muted, I believe.
Can you hear me now?
Yes.
Okay, thanks. Can you also see the PowerPoint?
Yes.
All right, good. So quick announcements to remind you, lab is online this week. As long as you have it done before your time to come to lab, you don't actually have to meet online for lab. You're just done for the week. But if not, just meet me at 2 o'clock online as usual. And then, like I said, it's an online lab. It really is easy. Speaking of lab reports were due in the past week, the 24th. But when I made that lab or that due date, I didn't take into consideration that you guys had spring break. So technically, I've just extended it until tonight. And also, as long as it's done before I get to grading it, to me, it's on time. And so, if you haven't done it yet, and I don't know about it, just get it done. What else? I guess that's it. Everything else is the same. No news. Don't forget to do independent work, even though you don't have to. It is extra credit. So for those of you who need a point boost, that would really help. So any questions? All right. This is going to be a big one. I've got to kind of back things up. I'm about to show you how to use a test cross to determine an unknown genotype. But before I do that, let me back up and show you what I mean by an unknown genotype. We were talking about a dihybrid cross, and we were talking about these dogs here. And when I say an unknown genotype, I mean this. Or even you guys, if you remember in the lab, when we talked about like cleft chin, like if you had a cleft chin, then you had the dominant phenotype, and we didn't know what your genotype was, right? So you had to write big D, and then the word or, and then big D, little d, because we didn't know. But you could have also just written that, right? Big D with a blank. Because if you have the dominant genotype, or this dog, for example, has the two dominant genotypes, that could be big B, or big B, little b. We don't know, right? That's what we mean by unknown genotype. So the next thing we're going to talk about is how to use a test cross to determine the unknown genotype. Now, I don't remember if there's any questions about this on the study guide, but I can tell you there almost definitely won't be on the exam. So read it a couple of times, but if you're studying for the exam and trying to memorize things, put this on one of the lowest priorities. So here we go, test cross. What is a test cross? It's a mating between an individual of a dominant homozygous recessive individual. So if anything, that would be the question. If anything, the question would be, who or what would you mate in a test cross? And the answer is obviously, first of all, the one with the unknown genotype. And you're going to mate them with somebody or something, with a homozygous recessive genotype. So again, for the exam, if anything, that's what you would need to know. And the next slide is going to show you why that is the case. Actually, I'm glad you came in. That reminded me, for attendance, for you four who are here, if you don't mind, send me an email whenever you get a chance. Let me know that you're here on time, if you can remember. If you don't remember and you remember late, that's fine. Yeah, there you go. And then for those of you online, we'll do the normal stuff. And the first word for attendance is rag, like something you would clean something with, for those of you online. Anyway, let's talk about this test cross. So again, we're using these dogs as an example. If you remember, a black lab, that is the dominant phenotype. Chocolate is the recessive phenotype. Now we know that chocolate lab, obviously we know both of their phenotypes, but we also know the chocolate lab's genotype. Because remember, if you have the recessive phenotype, that means you have to be homozygous recessive. The only way you can have a recessive phenotype is if you have both recessive alleles. Therefore, we know that puppy, that dog, is homozygous recessive. But again, when you have the dominant genotype, unless you've got some background information on the parents or the parents, then you don't know the genotype. So in this case, we don't know. We don't know if this dog is big B, or big B, little b. So to figure it out, we look at the possibilities of both, right? So here on the left, we look at this as if the dog was homozygous dominant. And then on the right, we look at this dog as if it was heterozygous, right? So this is both possibilities right here is what we're looking at. So we know if this dog was homozygous dominant and it mated with a homozygous recessive dog, then we know 100% of its offspring would be heterozygous. And I'll just do it really quickly. The full Punnett Square, because Punnett Squares are useful. As much as you might not be doing a test cross, you will be doing Punnett Squares. So let's just do one really quick. So your book only shows the one square, and you're going to get the same results here, but I just want to show it to you. Big B, little b, little b. So obviously 100% of them are going to be big B, little b. Does that make sense? Does anybody have any questions about that? You can all see how no matter what, if you have a homozygous dominant and heterozygous mating, then 100% of the offspring are going to be heterozygous. That's actually a test question too. That probably will be. It's definitely a question on the study guide saying, in what situation would 100% of the offspring be heterozygous? And the answer is when you mate a homozygous dominant and a homozygous recessive. So anyway, again, if this dog right here, if its genotype is homozygous dominant and we made it with that recessive phenotype dog, then we know all of the puppies are going to be black, right? And they're all going to have a heterozygous genotype. However, if this dog, if you look on the right here, is heterozygous, if we were to do the full Punnett Square, but we don't need to because I already showed you once, we would see that half of the dogs would be black and half of the dogs would be brown. Make sense? So that way, that's how you would do it. Now, let me ask you this. If we did mate this dog, this black dog, with this dog, and again, we don't know the genotype, we're trying to figure it out. Let's say it had, 4 puppies, and they were all black. Do we know for sure that this dog is actually homozygous dominant? If they're all black, do we know for sure? Or let me say it another way. Let's look at the other situation. If this dog and this dog mate, as in this situation, and 3 of them are black and 1 of them is brown, do we know for sure that dog is heterozygous? Any reason why?
Say it again.
If this dog, we're mating these dogs, and then let's assume we know that this dog, let's assume we get 3 black labs and 1 brown one. Do we know for sure that this dog is heterozygous as opposed to homozygous dominant? Anybody else? You said no? In this case, the answer is yes, because there's no way to get a brown dog unless you because remember, the brown dog, that's the recessive phenotype. So the only way you can get the recessive phenotype is if you have 2 recessive alleles, right? So we know for sure, if you get a brown puppy, for sure that black dog must have had a recessive allele. Otherwise, you wouldn't have had that. On the flip side, though, here I should have asked that first. If you were to do this, right, you mate this dog, and let's say you get all black dogs. Now, if you get all black dogs, that indicates it's probably homozygous dominant, but not 100%, because it's possible that this dog has recessive alleles and it just didn't give it to him during fertilization. Does that make sense? So we know if there's a brown that dog is heterozygous. But if they're all black, that does not necessarily mean it's homozygous dominant. That just means it didn't give a recessive allele. Now, granted, if you kept mating them and had hundreds of dogs and they were all black, eventually you could say, yeah, by the rules of probability, that dog must be homozygous dominant. But yeah, any questions about that? So again, just an example, and I'm probably not going to ask you anything about that on the exam. For the grand scheme of the semester, that's not a very important concept. Now, it is good to be able to do Punnett squares. That's going to be useful. Speaking of the rules of probability, let's talk about that. Your book points out that genetic crosses do obey the rules of probability, and these are the same rules that apply when you're tossing coins, rolling dice, drawing cards, playing the lottery, all that stuff. So, for example, if I flip a coin, what are the chances of it landing on heads? Good, 50-50, right? 1 and 2. If I roll a dice, what are the chances of it landing on a 5? Not that you guys need to know this. It's just examples. Just 1 and 6, right? No matter what the number is, it's 1 and 6. If I give you a deck of 52 cards and I say, all right, pull it. And what are the chances of you pulling out a red 2 of hearts? What are the chances of you pulling out a 2 of hearts? 1 and 52, right? Yeah, so all these rules are the same. Now, that brings us to the rule of multiplication, which is the probability of a compound event is the product of separate probabilities of independent events. Meaning, like you already said, if I flip a coin and it lands on head, the chances of that happening are 50% or 1 and 2. But if I were to say, all right, what are the chances of me landing on head and rolling a dice and it landing on, I don't know, a 2, right? And that's 1 and 6. But these are separate events. So if I were to say, what are the chances of both of those things happening, then you have to multiply it. So if you know anything about fractions, this is pretty simple. The top is the same. 1 and 1 is 1. 2 and 6 is 12. So the chances of rolling dice and landing on 6 and flipping a coin and landing on heads is 1 and 12 because those are separate events. Does that make sense? Whoops. And that brings us to this. So even though I keep showing you Punnett squares, this is nothing new. The only thing we've done now is added fractions to it. So we can actually talk about this mathematically. So I can't reach up there. When this female up top, that heterozygous female, when she goes through, she's going to go through meiosis, and those alleles are going to separate. So half of her eggs are going to be big B, half of her eggs are going to be little b. Same thing with the male. When he goes through meiosis, those alleles are going to segregate. So half of his sperm are going to have big B, half of them are going to have little b. So then the question would be, well, what are the chances then of one of her big B eggs matching up with one of his big B sperm? And then you do the Punnett square and you can see, all right, well, there's a 1 and 2 chance of this happening, right? And then a 1 and 2 chance of this happening. And if we're saying what are the chances of both of those things happening, then you have to multiply it. So 1/2 times 1/2 is 1/4. Or the easiest way to do it, again, is just do the Punnett square and look at it, right? So 1/4 are going to be homozygous dominant. 1/4 here for heterozygous, but another 1/4 here for heterozygous, and 1/4 for being homozygous recessive. But here's what your book didn't say. Either that or I left off the PowerPoint. I don't remember. So I just gave you the rules of multiplication. But then there's the rules of addition. So again, with the rules of multiplication, we're saying what are the chances of this thing happening and this thing happening independently, right? So when it's and, you multiply it. So this 1/2 times 1/2 is 1/4. But when you say what are the chances of this or this happening, when it's an or, then you would add it. So what's 1/4 plus 1/4? Feel free to say it online if you're good at math. Whoops. Yeah.
Is it 1/2?
Yeah. Well, yeah. 1/4 plus 1/4 is 1/2, right? So, and again, it makes sense visually when you look at this thing because there's 2 of these. It's hard to see. Where's my thing? There we go. 1/2. So, any questions about that? All right, so let me give you one more example, something you might get on the exam. Well, first of all, what are the chances of, I don't know, if you were to have a kid, what are the chances of it being a boy? Good, right? 50-50, right? So you might get a test question that says, what are the chances of this person mating with this person? Let's say we're talking eye color, right? Let's say this person has brown eyes. They both have brown eyes, and they're both heterozygous. So I might say, what happens when these 2 brown-eyed people mate? What are the chances of them having a girl with blue eyes, right? So we know the chances of having a girl is 1/2. And the chances of it being homozygous or recessive, which would be blue eyes, is right here. We can see right here, 1/4. So in this case, we would multiply. So 1/2 times 1/4 equals 1/8. So you might get a test question like that. So be careful. But there won't be many. This isn't a math class. So I do want to know that you can do these concepts. But also at the same time, I don't want you to have so many of these questions that if you're not good at math, it's going to drop your grade by a letter grade. So be prepared. Know that they're there. Hopefully I'll give you some more examples between now and then that you can do for independent work / extra credit. But that is what that is. Any questions about that? All right. Nice and easy. The second word for attendance is going to be eggs. Eggs is the second word for attendance. All right. Let's talk about family pedigrees. Again, we're going to go through this kind of quickly too. As far as the grand scheme of the semester goes, family pedigrees are not that important, so I'm not going to spend a lot of time on them. I don't know if there's going to be any questions about it on the study guide or the exam. If there are any on the exam, there's only going to be maybe one. Because again, as far as the semester is concerned, it's not a very important concept. So your book points out that Mendel's principles apply to inheritance of many human traits as well. So what we mean by that, to remind you, we're talking about Mendel, who's the person who figured all this stuff out. And he did all of his work with pea plants, right? He wasn't studying humans. So your book is just saying, so yeah, he was doing all this stuff with plants, but a lot of the stuff he worked with, they also apply to humans too. And you should know this because this is basically what we did for the lab. We looked at human traits that had this Mendelian ratios. But anyway, let's look at some of these things. Here's some examples. Again, you should be familiar with the last two, widow's peak and free earlobe. You know those from the lab. That's a human trait that's controlled by one gene, which is actually a pretty rare thing, and we'll come to that later. But these are some things that apply to which Mendel's rules apply. And also freckles. We didn't talk about freckles. Well, we talked about them, but they weren't officially on the lab. So having freckles is a dominant trait. Not having freckles is a recessive trait. These are just examples. I'm not going to ask you anything about that on the exam. Oh, yeah. Before we get to the pedigree, I said this during lab. I want to say it now. Hopefully you've got this message from lab. But the dominant trait does not imply that it's normal or more common. Because remember, especially for those of you on the Wednesday class, we went through, 3 or 4 phenotypes before we even got to one that was mostly dominant. Like how many of you in here have a cleft chin? Right? None of you, me included. My son's the only one in the room with a cleft chin. But that's a dominant phenotype, right? So again, just because it's dominant doesn't mean it's very, very common. Or freckles. I'm looking around. I don't think anyone in here has freckles. That's a dominant phenotype. So what is the one that's most common? It's called the wild type trait. You do need to know that. The wild type trait is the one that is most seen in nature. It could be dominant, could be recessive. Doesn't matter. Fact is, if it's the most common, it's dominant. And of course, this second bullet point, that should be rewritten because it should say this was demonstrated in the genetics lab because we did it already. Anyway, any questions about what the wild type trait is? All right, let's move forward. So when Mendel was studying those pea plants, remember from way back when we started this long chapter, we talked about how he cut off the male parts of the flower, and that way he could control. It was like, all right, this flower is going to breed with this flower, and he made sure that this flower bred with that flower. Remember all that? Now, we can't do that with humans, right? That's not ethical. I can't be like, I want to study height, so I'm going to get this really tall. Hey, you tall person, I don't need you to mate with that really short person, right? We can't do that. So how do geneticists study humans? Well, we use pedigrees, right? We look at matings that have already happened. Obviously, again, it's not ethical to make people have sex and study their kids, right? So we look at matings that have already happened. And that's what we're getting at to right here. Here we go. Here's a pedigree for attached ears. Now, I'm going to try to go through this quickly because, again, it's not that important, but I do want to show you at least once how to figure this out. So we know attached earlobes, that is what? The dominant trait. Actually, let me back up and make sure. We don't want to mess that up. Free earlobes, okay. The free earlobe is the dominant trait. I always forget that because, again, it's not important. It's just an example. So free earlobes are the dominant trait. So we know big F, good grief, or big F, little f, That's free earlobes, right? If you can read it with all that garbage. So that way, this means we know that if you have the attached earlobes, that is the recessive trait. Little f, little f. So let's really quickly go through this. We're going to try to figure out the genotype of all these people. How about these people in purple then? All these people with attached earlobes. What is their genotype? And again, they have attached earlobes, which is the recessive phenotype. So pedigrees aren't important, but what I'm asking you right now is an important concept. You should know that if they have the recessive phenotype, that their genotype is what? Well, yeah, definitely FF. But what are we talking? Big F, big F, big F, little f, little f, little f. Yep. Oh, and speaking of which, not that this is that important either, but the box in a pedigree, a box just means male, circle means female. But here's a hint. I don't know if you guys aren't paying attention or I'm going too quick, but I'm asking you what the phenotype, the genotype is, the people in purple, right? I've already written it right here. Little f, little f, little f, little f. We know, and again, the pedigree is not that important, but what I'm saying right here is important. So if you're taking notes, write this down. If someone or something has the recessive phenotype, that their genotype has to be homozygous recessive. The only way to have the recessive phenotype is if you have both of those recessive alleles. So anyway, that being said, let's move forward. I'm going to go ahead and write that down. We know anybody in purple is little f, little f. It's going to get sloppy. I might not be able to do all of them. Otherwise, it'll get too sloppy. Now, let's see. Knowing that these people are little f up at the top right. How am I going to put this? Okay. Here's where we'd have to do Punnett squares. And Punnett squares, again, pedigree is not that important. Punnett squares you need to do. So let's try to figure out this person's genotype and this person. We're basically going to do a test cross, right? Because remember, test cross is crossing somebody with an unknown genotype with a recessive phenotype. And that's what that is. So really quickly, let's do that. Up at the top, I'm going to put that little f, little f. Little f, little f. Now let's talk about the two possibilities. Let's first try, I don't know, homozygous dominant, right? Because we know that this person over here is big F and then blank, right? Big F something. So let's see if it's big F, big F. If we have big F, little f, if we have homozygous dominant and homozygous recessive together, as you might remember from the test cross thing we just did, 100% of the offspring would be big F, little f, right? Does that make sense? You guys with me on that? If you miss a homozygous dominant with a homozygous recessive, 100% of the offspring are going to be heterozygous. So the question is, are 100% of their offspring heterozygous? Are all those heterozygous? You guys must be still on spring break. I'm asking you if 100% of these offspring are heterozygous. Nobody? For extra credit, read the thing that says homozygous recessive. All right. So no, they're not 100% heterozygous because we see right here, this one's, again, homozygous recessive because they have the recessive phenotype. So we know for a fact that this person right here at the top right, if I can get it working. Yeah, there we go. This person right here has to be big F, little f. Again, the pedigree is not that important, but I am kind of disappointed you guys aren't seeing these things that are right in front of your face when I'm asking if 100% of these people are heterozygous. And literally, my finger's touching somebody who's homozygous recessive. And I was just talking about how they have to be homozygous recessive because they have the recessive phenotype. So please study up. But if you took the break off, like actually took the week off, I understand your brain might be a little bit foggy, but we'll move forward. Clearly, if that one's not making sense to you, then there's no need for me to go through all the rest of them and just confuse you even further. So we're going to move forward. Hopefully, if I have time, I'll come up with some phenotypes, some pedigrees to share with you and that you can do for extra credit if you want. Anyway, actually, before I move forward, are there any questions about this pedigree? All right, here we go. This is where we get a little bit more realistic and a little bit more important. Now we're going to talk about human disorders. So, like, really, right? Who cares about freckles and cleft chin and free earlobes? That stuff's not really that important, right? That's the superficial stuff. So let's actually talk about disorders, diseases. In this case, we're going to talk about human disorders caused by a single gene, which, as you're going to learn later, is pretty rare. Some disorders are known to be dominant or recessive and controlled by a single gene located on autosomes. So to be clear right now, again, what we're about to talk about, these are cases of simple dominant or recessive. And later, you're going to hear that there's other versions of this. But for now, I just want to be clear. Right now, we're talking about either or. And these are also controlled by a single gene. And again, later, you're going to realize that that's not quite normal. And then again, one last thing to note, we're talking about things on autosomes. So these are not sex chromosomes, right? These are all the other 22 pairs of chromosomes that we're talking about. And these are just examples. I think there's questions about this on the study guide, but I'm not going to ask you to remember any of this stuff. Yeah, there's a table. I think it's from your book. If not, that's okay. Again, these are just examples. These are some recessive disorders, meaning if you have this, if you have the recessive disorder, that's another way of saying you have the recessive phenotype. In other words, if you have one of these disorders, that means you have the homozygous recessive genotype. And then, of course, here's some dominant disorders, which is the opposite. That means you have either homozygous dominant genotype or you have a heterozygous genotype, with an exception. And I'll come back to that exception. So when you're studying for this, read this if you want, but there's nothing to memorize on this table. Let's talk about them. First, we're going to talk about recessive disorders. Most disorders, the kind we're talking about here, are recessive. So there's more recessive disorders than there are dominant disorders. And that makes sense because we just saw this list, right? There's a bigger list up top for the recessive than there are dominant. Any guesses to why there are more recessive disorders than dominant? And let's just say most of them are deadly. That's a hint. Most of them are deadly. Well, I'll tell you, the reason why we have mostly recessive, because in order to have the disease, you have to be, I'm just going to use F again since we were already using it, you have to be homozygous recessive. To have this disease, because it's a recessive disorder, you have to have both recessive alleles. So if it was deadly, you would have to have both of those for it to kill you. However, and we're going to talk about this later in more detail when we talk about dominant disorders, if it was a dominant disorder, how many dominant alleles do you need to have the dominant disorder? Just one. I think somebody said it. You just need one. So obviously, a lot of people are going to be dying from the dominant disorders. Therefore, it's going to be less common in the human gene pool because more people are going to die from it. Anyway, we'll come back to it. Let's talk about these recessive disorders. Nah, we'll skip that. Cystic fibrosis is one example. It is the most common lethal genetic disease in the United States. That's probably why your book included it. And even though it is the most common lethal disease in the genetic disease in the United States, I'm not going to ask you anything about it on the exam. This is just an example. But we'll talk about it anyway. It's caused by a recessive allele that is carried about 1 in 31 Americans, which is, again, the most common in the United States. So that's all we're going to talk about as far as recessive disorders. And again, cystic fibrosis is just an example. I think there are some questions about it on the study guide. There won't be any questions about it on the exam. You can look it up for independent work if you want, because notice I didn't say anything about it other than the fact that it's the most recessive. I didn't tell you what the symptoms are or how old you are when you die, nothing like that. Dominant disorders, really quickly. Again, we're talking about disorders here, genetic disorders. Some of them, the last ones we talked about, are caused by recessive alleles. These are caused by dominant alleles. Chondroplasia is a good example, and I talked about this in lab. It is a form of dwarfism. But here's an interesting case, and you don't need to write any of this down. I'm not going to ask you anything about this on the exam. This is one of those weird situations where it's not equal. Meaning, if you are homozygous dominant, well, you're not. Because if somebody like, somebody who's carrying the allele mates with somebody else who's carrying the allele, and they come together and they have a zygote that's homozygous dominant, that thing's not even born. So there is nobody alive that has the homozygous dominant genotype because it's deadly, like so deadly you're never even born. So anytime you see a dwarf, at least this form of dwarfism, that means they are heterozygous, right? They are big A, little a. So a person who has a chondroplasia has a 50% chance of passing that condition on to their children. And I'll show you what I mean when I show you the next slide, because we'll look at a Punnett Square. Actually, I'll just do it now. Are there any questions about what's on the slide right now? All right. So here's what we mean. If you have a dwarf, as you can see on the right-hand side of the screen here. All right, so let me back up. There is a study guide question about this, and there might be a test question. But if there is a test question, I'm not just going to require you to memorize all the ins and outs of a chondroplasia. I'm not going to require you to remember that homozygous dominant is deadly and only the heterozygous are the dwarfs. I'm not going to require you to remember that. If I give you a question about this, all that information is going to be given to you on the exam. But I might say that. I might give you all that background information and say, all right, if somebody, if a dwarf mates with a normal person, or by normal, I mean they don't have a chondroplasia, I might say, what are the chances of their kid having dwarfism? And then you could do a Punnett Square, because you know if the person is a dwarf, that their genotype has to be heterozygous, right? Because if they were homozygous dominant, they would be dead. If they were homozygous recessive, they wouldn't have dwarfism. Speaking of which, we know that their mate, who does not have dwarfism, has to be homozygous recessive. So all that information should be enough for you to do this Punnett Square. And then you could do this and figure out, okay, there is a 50% chance that they would have a dwarf for a child. Does that make sense? You might get a question like that on the exam. And to make it harder, like I was saying earlier, I might throw in something for the rules of probability. So I might say, what are the chances that they will have a boy with a chondroplasia? So a boy is 1/2. A chondroplasia, 1/2. So 1/2 times 1/2 is 1/4. So be prepared for that question on the exam. Also be prepared for the wording, because I might ask you, I might give you this and say, what are the chances of them being homozygous dominant? And you can see there's a 0% chance of that. So again, when you're studying and you do this question on the study guide, don't memorize that question, because I will change the numbers up so things will be different on the exam. Any questions about that? All right, the next word for attendance is going to be watch. Like the thing on your wrist, if it's a noun, or if you're looking at something, you are watching it, the verb. All right, another disorder here, a dominant lethal disorder. Oh, excuse me. This is what I was getting at earlier. Dominant lethal disorders are much less common than lethal recessive disorders. And I already talked about why. Again, if to have this disorder, you only need one of the alleles. Therefore, it would make more sense that it'd be easier to get. Therefore, it's going to be less common because people are dying from it. Huntington's disease is an example. That's a degeneration of the nervous system that usually doesn't begin until the middle age. Until middle age. When I said it like that, it sounded like I was talking about a period in history. Once the deterioration begins, it's irreversible and inevitably fatal. And because the allele for Huntington's disease is dominant, any child born to a parent with the allele has a 50% chance of inheriting it. So if you have this disease, you have a 50% chance of giving it to your child, regardless of your mate. Plus, and the tricky thing about this, well, it's not so much anymore, but back in the day, imagine when they didn't know about genetics, you're completely normal until middle age. So back in the day, unless you knew, you might just think you didn't have it. In middle age, back in the day, people were popping out kids early and a lot, right? So you might be having a lot of kids before you're like, oh my God, I've got Huntington's disease. Well, too late, because you already had 6 kids, right? Anyway, Huntington's disease, again, just an example. I'm not going to ask you any questions about that on the exam. All right, every chapter has this, the process of science. And as always, we are going to skip it. As always, I highly recommend reading it. It's good for your brain to think scientifically. There's probably questions about it on the study guide. There won't be questions about it on the exam. We will skip through. And of course, as you read it, or after you read it, if you have questions, let me know. Genetic testing, here we go. We're still talking about disorders here, human disorders. So let's talk about genetic testing. There are tests that can detect disease-causing alleles. And this would be very important, right? If you have something or you're mating with somebody and you want to know if they have it because that really increases your chance of your kid having it, then this is where this would be important. Actually, before I even get into this, I'm going to let you know none of this is going to be on the exam either. So anyway, one of the tests you can do is the amniocentesis. Basically, you stick a needle in, you pull some liquid out, you test that liquid, and there you go. And again, I'm going through this quickly because it's not going to be on the exam. It's really not important in the grand scheme of the semester. So, does anybody know what's going on right here? What do you call this thing? What are they doing?
Surgery?
It kind of looks like surgery because they're all geared up. It's not.
Is it some ultrasound?
Yeah, exactly. It's an ultrasound. So you can see you can see the baby. That's what they're doing there, right? That thing allows you to see the baby, which makes sense because it's kind of hard to see is right here is the needle, right? So they're about to jab her in the belly. So it would be helpful to see where the baby is so you don't jab the baby too. That's all that is. Yeah, there's a picture of what we're talking about. Anyway, again, for the semester, for the exam, this is not important. Then there's CVS. It's a newer way of doing it. I'm not even going to talk about it because, again, not important for this grand scheme of the semester. All right. We just finished the first main bullet point of Chapter 9. Chapter 9 is a doozy. So let's jump into some stuff. We're going to talk about variations on Mendel's laws. So to remind you of what we talked about so far. What we've talked about so far is not very common. Everything we've talked about so far is one gene controlling one character, and it's either completely dominant or completely recessive, right? So like chin dimples and eye color and freckles and the color of a pea flower, right? It was either this or that, and there was only one gene that caused it. It turns out I've almost been wasting your time because most things in nature aren't like that. Think about height, for example, right? If height were that way, there would only be two sizes of humans, tall and short, right? You would either have, 6'6" or 4'6", right? And nowhere in between. But it's not like that. It's more complicated. So let's talk about these variations on Mendel's law. The rules of probability still exist. These are still valid for all sexually reproducing organisms. But again, like I was saying with height, that's a good example. The rules I just taught you that I just spent so much time talking about, that doesn't apply to things, everything. Like again, height is a good example. So mostly Mendel's rules cannot account for patterns of inheritance. And that's what we're about to talk about. Things that are more realistic and less like what I just talked to you. So for the exam too, before I jump into this is how the questions are going to go. I'm going to give you some mating. I'm going to say this thing mated with this thing, and this was the results. And then I'm going to ask you, was that a case of complete dominance, which is what I've been teaching you earlier, the Mendel stuff, like freckles, eye color, hitchhiker's thumb, all that. I'm going to say, was it complete dominance? Was it incomplete dominance? Was it, I'll come back to that one. Pleiotropy, polygenetic inheritance, or epigenetics. So as I'm teaching this, keep that in the back of your mind. That's how the test questions are going to go. I'm going to give you an example. This thing mated with this thing. Here's the results. Which of these is it? All right. So for example, if I said a person with brown eyes mated with another person with brown eyes, 75% of their kids had brown eyes, 25% of their kids had blue eyes. What is that an example of? And you would say that is an example of complete dominance. That's the stuff we've already talked about, right? Where it's either this or that. So here we go. Let's talk about some other examples. Incomplete dominance. This is when the F1 phenotype falls between the two parents. So to remind you, the stuff we talked about earlier wasn't incomplete dominance. It was complete dominance. You were either a purple flower or you were a white flower, right? Or in that case, you were always purple because purple was dominant. You're either brown eyes or you're blue eyes. And again, it would usually be brown eyes because brown is dominant. Incomplete dominance, it falls in between. Here's some examples. I don't remember if these are from your book or not. But if I give you on a test question, all right, we made it a white flower with a red flower, and some of the offspring were pink, right? Pink falls between red and white, right? That's why it's called incomplete dominance. So if you get a test question like that, that would be the answer. Now, meanwhile, if I said we bred a white flower with a red flower and the offspring were red, then you'd be like, oh yeah, that's complete dominance. But if I say it's pink, that means it falls in between. That would be incomplete dominance. Another example, if you breed a black mouse and a white mouse and you were to get a gray mouse. Again, what comes between black and white? Gray. That's how you know it's incomplete dominance. Now, if I was talking on the exam and I said I bred a black mouse and a white mouse, and we got 75% black mice and 25% white mice, then you would say, yeah, that's complete dominance, right? Because it's either black or white. In this case, it's in between the two. And then finally, another example could be curly hair with straight hair, and you get what's in between curly and straight, which is wavy. So any questions about incomplete dominance? Again, the key to recognizing this is it falls between the two phenotypes. That's it. It should be a pretty simple concept. Oh, another thing to say. Most likely on the exam, I'm going to make some stuff up. I'm not going to give you a real example of incomplete dominance or complete dominance. I might say something like, I don't know, a red alien and a purple, red alien and a blue alien mate, and their offspring are purple, right? And if, I don't know if any of you guys are art majors, but what happens when you mix red and blue? What color do you get? Purple, right? So if that were to happen, you would be like, oh yeah, that's incomplete dominance because it falls in between those two phenotypes. So let's see it in action. Here we have a purebred red flower, right? Homozygous dominant, mating with a purebred white flower, homozygous recessive. Obviously, when the red one goes through meiosis and those R's segregate, it doesn't matter that they segregate because 100% of them, 100% of the sperm or egg are going to be big R. Same thing with the white one. 100% of the sperm or egg will be little r. Now, when you mix those things, obviously, 100% of the offspring are going to be heterozygous, big R, little r. And because this is an example of incomplete dominance, the genotype falls in between the two and we get pink. And then, of course, if you were to mix those two, you would get the same numbers. You would get 1/4 homozygous dominant, you would get 1/2 heterozygous, and you would get 1/4 homozygous recessive. Except now the only difference is, again, homozygous dominant, it's still red. But again, the heterozygous are going to be the mixture of the two phenotypes. So again, that's the key. Anytime there's a mixture of the two phenotypes, then you know we're talking about incomplete dominance. And of course, if you look at the genotype ratios, those are still the same. We're still looking at a 1 to 2 to 1 genotype ratio, just like we used to with the complete dominant situation. But now we also have a 1 to 2 to 1 phenotype ratio. But again, that might sound complicated. The most important thing you need to know is that the phenotype falls between the two. So any questions about that? All right, your book gives a real example. I'm not going to ask you any questions about it. Hypercholesterolemia. It's basically when you have really high blood pressure, which could be deadly. How deadly? Your book points out that sometimes people have heart attacks as early as the age of 2. If you can imagine that. How does it work? Well, if you're homozygous dominant, you have a normal cell. You have all these things on the outside of your cell that take away the cholesterol out of your blood system. If you have the disorder completely, that means you're homozygous recessive. Therefore, you're not making any of those proteins. Therefore, you're not taking any cholesterol out of your blood system. Therefore, it's really deadly. But if you're heterozygous, here you've got one allele that works and says, hey, make this protein. And of course, the other one doesn't. So you still are making some of those proteins that pull the cholesterol out of your bloodstream, but you're not making it a lot. Again, this is just an example. There'll be no questions about this on the test. There might be some on the study guide. The key here is the homozygous dominant. See that phenotype right here? That's a lot of proteins pulling it out, right? The homozygous recessive, that phenotype is there's no proteins. So again, the key to seeing the incomplete dominance is that the phenotype for the heterozygote is in the middle, right? It's not a lot of protein like the homozygous dominant. It's not no protein like the homozygous recessive. It's right there in the middle. So in the eye color, again, eye color is an example of complete dominance. But if it was an example of incomplete dominance, then instead of having, if you had a brown-eyed person and a blue-eyed person, then you might have an offspring with like half brown, half blue, or not half brown, half blue, but some mixture of the colors. All right, any questions about that? So again, that was incomplete dominance. The next thing we're going to talk about is the Abo blood groups. I'm going to go through this pretty quickly too. And to let you know, even though I'm teaching you about the blood groups themselves won't be on the exam. And I'll tell you what will be when we get there. Abo blood groups. I'm sure you guys have heard of this. How many of you guys know your blood type? Like I'm O positive. Does anybody know what their blood type is? Doesn't matter. But here we go. Let's talk about what it means. I'm sure you've all heard of it. How many of you know what it means? I didn't. Yeah, it basically dictates, like you said, who can share, right? Like, for example, I'm about to teach you why. As a O blood type, I can share my blood with anybody, but which is good for anybody. But the bad thing for me is I can only have blood from the O blood type, and we'll see what we mean by that. What we're talking about, before I even get into it and start showing you pictures, what we're talking about is a carbohydrate. So blood type A means you have the A type carbohydrate on the outside of your blood cells. Blood type B means you have the B type carbohydrate on the outside of your blood cells. AB means you have both of those type of carbohydrates on the outside of your blood cells. And O means you have none of those carbohydrates on the outside of your blood cell. So this looks complicated, but I'm going to go through it quickly because, again, I'm probably not going to ask you. So what we're looking at here, this is really, I don't even like this example, and you'll see why when we talk later. But anyway, like I said, you either have carbohydrate A on the outside of your blood cell, you have carbohydrate B, you have both, or you have none, right? That's A, B, or O. And basically what this means is if you're carbohydrate A, that means that's it. You are anti-B, right? If you are A, you don't want B. So that's why if you are blood type A, you can have anything other than B blood, because it's got that B carbohydrate, and your blood hates B. And you also can't have AB, because again, even though it has the A, which is what you are, it has the B, so that's bad. Same thing said for the B blood type. If you're B blood type, you're anti-A. Therefore, obviously, A blood type is bad for you, but also so is AB, because again, it has the A. Here's what we have, the AB. These people are interesting. These are like the opposite of me, because they're not anti-anything. They have the A type, they have the B type, therefore they're not, they're not discriminating. They are the universal acceptor. These people can get blood from anybody. Meanwhile, this is me, O type. I don't have the A carbohydrate. I don't have the B carbohydrate. So my blood hates. I've got hateful blood. It doesn't like A, it doesn't like B. So anything would kill me other than the O blood type. And again, I would take questions and talk about that longer, but that part won't be on the exam. What's important here, and I don't really like this as an example, but your book uses it, so do I. But what we're talking about here is this. This is the important part right here. This is an example of codominance. So again, if you're keeping track, the first thing we've been talking about for the first half of this chapter was complete dominance. The thing we just got finished talking about with the pink flowers, for example, was incomplete dominance. And here's where it gets tricky. Now there's a third one called codominance. So codominance is where you have both phenotypes, right? So again, if you were to mate a red flower and a white flower and it was pink, that's incomplete dominance. That's what we talked about earlier, because it's a mixture of the two. Now, if you were to mix red and white and you have a red flower with white spots or a white flower with red spots, that is codominance. So codominance is both. So again, using my hypothetical alien example, if I mate a red alien with a blue alien and the offspring are all red with blue spots, then that would be codominance because it's both phenotypes. Make sense? It's both phenotypes. It's not a blending of them like incomplete dominance. It's both. And that's why I don't like the blood type as an example, because not only with the blood type are we talking about codominance, which is a new concept for you people, it's also a situation where you have four different phenotypes. And I didn't even want to jump into that yet, but again, that's what the book does. When we get back, we'll talk about we can do this one really quick. Pleiotropy, really quickly. We can do this very quickly. Pleiotropy is simply this. One gene, but several characteristics. So everything we've talked about so far is one gene, one characteristic. This gene for eye color, this gene for blood type, this gene for cleft chin. But now we're saying one gene, many characters. Your book gives the example of sickle cell, which I'm not going to ask you about because it's just an example. But the thing is, if you have that one gene, it gives you that disease, and then you have this long list of symptoms. So again, the key to pleiotropy is one gene and then many different characters. And that's where we'll stop. And when we get back on Wednesday, we'll talk about the opposite of that, which is polygenic inheritance. And the last question, the last word for attendance, let's just say basis, since it's right here. Basis. All right. Anybody have any questions? All right. I might see you online for lab today. Otherwise, I'll see you on Wednesday.
Okay, have a good one.
You too.